G-protein coupled receptors, commonly known as GPCRs, are one of the most important families of cell surface receptors in the human body. They play a vital role in transmitting signals from various extracellular substances such as hormones, neurotransmitters, and local mediators into the cell to produce a specific physiological response. These receptors are also referred to as 7-transmembrane receptors because they consist of a single polypeptide chain that crosses the cell membrane 7 times, forming 7 alpha helical segments. Structurally, GPCRs have 3 distinct regions: an extracellular domain, which usually serves as the binding site for ligands, a transmembrane domain, made up of 7 hydrophobic helices that traverse the lipid bilayer, and an intracellular domain, which interacts with special signaling proteins known as G-proteins. These G-proteins are heterotrimeric molecules consisting of 3 subunits: alpha, beta, and gamma. Among these, the alpha subunit binds guanine nucleotides and has intrinsic GT-PASE activity, while the beta and gamma subunits usually remain together as a functional dimer. In the resting state, the G-protein remains bound to GDP and is attached to the receptor on the inner side of the plasma membrane. When an agonist or ligand binds to the GPCR, the receptor undergoes a conformational change that activates the G-protein. This activation causes the exchange of GDP for GTP on the alpha subunit, leading to the separation of the alpha subunit from the beta-gamma dimer. Both components can then interact with various effector proteins such as adenylyl cyclase, phospholipase C, or ion channels, thereby generating second messengers like cyclic AMP, inositol triphosphate, diacylglycerol, or intracellular calcium ions. Depending on the type of G-protein coupled to the receptor, different intracellular pathways are activated. The GS type stimulates adenylyl cyclase and increases CMP levels, as seen with beta and beta-2 adrenergic receptors. The GI type inhibits adenylyl cyclase, reducing CMP formation, and is involved with alpha-2 adrenergic, muscarinic M2, and opioid receptors. The GQ type activates phospholipase C, which hydrolyzes membrane phosphatidylinositol into IP3 and DAG, leading to increased intracellular calcium and activation of protein kinase C. This is the mechanism behind alpha adrenergic and M3 muscarinic receptor actions. Thus, GPCRs can control a wide range of cellular activities including metabolism, secretion, muscle contraction, and neuronal signaling. The signaling process is terminated when the alpha subunit hydrolyzes GTP to GDP, thereby returning to its inactive form and reassociating with the beta-gamma dimer to reform the resting heterotrimeric G-protein complex. Prolonged stimulation of GPCRs can lead to desensitization and downregulation. This occurs when the activated receptor is phosphorylated by specific G-protein, coupled receptor kinases, and then binds to beta-arrestins, which block further G-protein interaction. Over time, the receptor may be internalized into the cell by endocytosis, either to be degraded or recycled back to the cell surface. This mechanism explains the tolerance that develops to drugs such as beta-agonists and opioids upon repeated use. Clinically, GPCRs are of immense pharmacological importance. Nearly half of all modern therapeutic drugs act by targeting these receptors. For example, beta blockers such as propranolol antagonize beta-adrenergic GPCRs to reduce heart rate and blood pressure. Salbutamol, a beta-2 agonist, relaxes bronchial smooth muscle in asthma. Clonidine activates alpha-2 adrenergic GPCRs to reduce sympathetic tone. Atropine blocks muscarinic GPCRs to reduce secretions and induce bronchodilation. And morphine acts on mu-opioid receptors coupled to GI proteins to produce analgesia. In summary, G-protein coupled receptors are the most versatile and widespread signaling systems in the body. By linking extracellular signals to intracellular responses through the activation of G-proteins and second messenger cascades, they regulate key physiological functions ranging from cardiac output and vascular tone to sensory perception and behavior. Their ability to amplify signals and their complex regulation make them a central focus of pharmacological research and a major target for drug development.